Microfluidics-based immunoassays for biomarker detection are revolutionizing disease diagnosis by making tests faster, more accurate, and cost-accepting compared to traditional laboratory methods. But why is this important, and how does it work? Immunoassays are tests that detect specific molecules, such as proteins or antibodies, in biological samples like blood, saliva, or urine, which serve as biomarkers for diseases. In traditional lab tests, large sample volumes, expensive reagents, and complex equipment are required, leading to high costs and long waiting times for results. Now, imagine a tiny lab-on-a-chip that can process a drop of blood in minutes, detecting diseases instantly. Have you ever taken a rapid COVID-19 test? That's a simple form of an immunoassay, but microfluidic platforms take this concept further by precisely controlling small fluid samples in microscopic channels, integrating advanced detection methods such as fluorescence, electrochemical sensing, or nanoparticle-enhanced assays. For instance, researchers have developed a microfluidic device that detects cancer biomarkers in just a few drops of blood, giving results in under 15 minutes. An innovation that could transform cancer screening, especially in remote or low-level areas where traditional diagnostic labs are unavailable. Similarly, microfluidic immunoassays are being used to detect heart attack indicators such as troponin proteins within minutes, allowing doctors to quickly confirm a heart attack and begin immediate treatment, potentially saving lives. Another fascinating example is in infectious disease detection. Microfluidic chips have been developed to rapidly identify bacterial infections and even detect antibiotic resistance, helping doctors prescribe the right antibiotics without unnecessary delays. What if a single test could check for multiple diseases at once? This is already happening with multiplex microfluidic immunoassays, which can simultaneously detect markers for diseases like HIV, tuberculosis, and malaria from a single drop of blood, making diagnostics more efficient and cost-efficient. Researchers are even exploring microfluidic devices for at-home testing, where a person could monitor chronic diseases such as diabetes or kidney disease using a portable chip connected to a smartphone. Imagine being able to test yourself for flu, COVID-19, or even early-stage cancer with a simple home device, sending results instantly to your doctor. With continued advancements, microfluidic immunoassays could soon make personalized medicine a reality, allowing people to track their health in real time and receive tailored treatments based on their biomarker profiles. Microfluidic immunoassays utilize miniaturized fluidic channels to detect biomarkers with high precision, relying on antigen-antibody interactions where target molecules bind to specific antibodies immobilized on a surface. This binding triggers measurable responses such as fluorescence, electrochemical changes, or color shifts, enabling real-time detection with minimal sample volumes. The miniaturized nature of microfluidic channels allows for controlled sample handling, reducing errors, increasing sensitivity, making these platforms ideal for detecting disease markers in blood, saliva, and urine. COVID-19 rapid antigen tests use microfluidic immunoassays to detect viral proteins, providing quick and accurate diagnoses without laboratory infrastructure. These immunoassays work like a fingerprint scanner, precisely detecting unique molecules to confirm the presence of a disease. Compared to conventional enzyme-linked immunosorbent assays, ELISA, microfluidic immunoassays require smaller sample volumes, lower reagent consumption, and provide faster results. Their high sensitivity enables early disease detection, while their integration with lab-on-a-chip technology allows for automation and multiplexing, detecting multiple biomarkers simultaneously. These benefits make them particularly valuable for point-of-care diagnostics in resource-limited settings, where rapid and accurate testing is critical. Microfluidic devices for tuberculosis detection allow healthcare workers in remote areas to diagnose patients within minutes instead of waiting days for lab results. These assays are like self-checkout kiosks, um, automating the process and speeding up results while reducing costs and errors. There are various microfluidic immunoassay formats, including lateral flow assays, bead-based assays, and digital droplet microfluidics. Lateral flow assays, such as pregnancy tests, use capillary action to move fluid along a strip, while bead-based assays employ tiny magnetic or fluorescent beads to enhance signal detection, making them highly sensitive. Digital microfluidics, which manipulates discrete droplets on a chip, offers precise control and automation for high-throughput screening. Each approach provides unique advantages depending on the required sensitivity, portability, and scalability. Magnetic bead-based microfluidic assays are used in cancer diagnostics to detect tumor markers in blood samples, enabling early intervention. These different types of assays are like different coffee machines. Some are instant and simple, while others are high-end and programmable, digital microfluidics. Microfluidic immunoassays play a crucial role in detecting infectious diseases such as malaria, HIV, and influenza by identifying pathogen-specific proteins or antibodies in bodily fluids. Their speed and accuracy make them invaluable for early diagnosis and epidemic control, and their enclosed microfluidic environment reduces the risk of contamination, ensuring highly reliable results even in non-sterile conditions. Malaria detection kits using microfluidics can identify the parasite in a blood sample within 10-15 minutes, significantly reducing diagnostic delays in endemic regions. These tests function like smoke detectors, quickly alerting to the presence of a potential health threat before it spreads. Microfluidic immunoassays are often integrated into lab-on-a-chip LOC platforms, which automate sample processing, biomarker detection, and data analysis within a single portable device. These self-contained systems eliminate the need for extensive laboratory infrastructure, making point-of-care diagnostics accessible to remote and underdeveloped regions. Some LOC devices are even compatible with smartphones, enabling rapid telemedicine-based healthcare solutions. Portable lab-on-a-chip devices for HIV monitoring help track viral load levels in patients, allowing real-time treatment adjustments in low-cylinder settings. A lab-on-a-chip is like a multi-tool gadget, combining multiple laboratory functions into a compact and user-friendly device. Microfluidic immunoassays are widely used in oncology for detecting circulating tumor cells and biomarkers like HER2, PSA, and CEA. These devices facilitate non-invasive cancer screening, personalized treatment monitoring, and early-stage diagnosis, significantly improving survival rates. By continuously tracking biomarker levels, they allow oncologists to adjust treatments in real time based on a patient's response to therapy. Liquid biopsy microfluidic platforms detect cancer markers in a simple blood test, offering an alternative to invasive tissue biopsies. These assays act like precision-guided drones, continuously monitoring and targeting specific cancer markers with high accuracy. Emerging applications of microfluidic immunoassays include diagnosing neurodegenerative diseases such as Alzheimer's and Parkinson's by detecting specific biomarkers in cerebrospinal fluid or blood. The ability to measure proteins like tau and beta amyloid in real time provides early diagnostic advantages, potentially enabling preventive interventions before irreversible neuronal damage occurs. Microfluidic devices detecting tau proteins in blood samples have shown promise in diagnosing Alzheimer's years before symptoms appear. These assays function like early warning systems, detecting neurological disease markers before significant damage occurs, much like sensors predicting earthquakes. Despite their advantages, widespread adoption of microfluidic immunoassays faces challenges such as fabrication complexity, reproducibility, and regulatory approval. The precision engineering required for microfluidic devices increases production costs, while ensuring consistency across different batches remains a technical hurdle. Additionally, meeting medical regulatory standards requires extensive validation, which can delay commercialization. Addressing these challenges is crucial for integrating these technologies into mainstream healthcare. Many promising microfluidic cancer diagnostic platforms remain in experimental stages due to high costs and prolonged regulatory approval processes. Developing and approving microfluidic immunoassays is like launching a new spacecraft, requiring precision, reliability, and rigorous safety checks before deployment. The future of microfluidic immunoassays lies in integrating artificial intelligence, AI, nanotechnology, and Internet of Things, IoT, connectivity. AI enhances diagnostic accuracy by analyzing vast data sets, while nanomaterials improve biomarker detection sensitivity. IoT-embedded devices facilitate real-time remote health monitoring, revolutionizing personalized medicine and preventive healthcare. These advancements will likely make diagnostics more accessible, efficient, and cost-effective in the coming years. Next-generation microfluidic platforms with AI-driven analysis are being developed to detect early-stage sepsis, dramatically improving patient outcomes in emergency care. Future microfluidic immunoassays are like smart assistants, continuously learning and evolving to provide better, faster, and more reliable health insights.